Hello, welcome everyone. It is Talktoptera 3 on September 4th, 2025, which is 3 to the second power, 2 to the second power, 45 to the second power. Couldn't ask for a more auspicious context to speak with Daniel Lanino Bilezikian, who will be discussing a variety of topics from the Centriolic to the sentient and artistic. So without further ado, to you, Daniel, and we will look forward to this presentation and discussion sequence. Thank you.
Great. Thank you, Daniel. I hadn't realized you were such an auspicious date, but what a perfect day to start. Nice to see you as well, Tucker. So today we're going to be going over a brief overview of my past few years of work, which are generally focused around the foundations of consciousness. So the substrate of consciousness is where I sort of focus. I come from a material science background, so I'm interested in the substrate of consciousness as a real material phenomena and how it interacts with the collection of materials that make up our organic body as well. So we're going to start by not going directly to the body or the organism as a whole and try to motivate ourselves from why we should even think about quantum mechanics being related to consciousness at all. And in order to do this, we're going to discuss a field that I've termed sentient wave mechanics. And this is a emerging idea in its formalized form, but it's been around for quite a long time since the founders of quantum mechanics. Max Planck and Schrödinger famously held views similar to this, but quantum information hadn't really matured to the point where they could formalize it yet. However, today, quantum information has progressed significantly since their day, and we're going to get to see why many people are starting to connect quantum theories to theories of consciousness. Now, we'll start off with a interesting individual for the future development of our world who is following a similar path, specifically Hartmut Neven and Google Quantum AI, where they're exploring very similar ideas. Their ideas are directly inspired by orchestrated objective reduction, as put forward by Penrose and Hameroff several decades ago, but diverge in the details and are a bit agnostic about the biology currently. They're more interested in approaching the idea of consciousness from a purely quantum information theoretic view. And they're looking to combine this with an experimental testing paradigm to see where they can find these effects in biology and whether or not they can expand them with brain-computer interfaces in order to create expanded sentient experiences. Now, their proposals are driven by several quantum correlates of sentience, where if we look at the fundamental laws and theorems and axioms defining quantum mechanics, and the same theorems and axioms that define conscious states, we see some really interesting correlations between the two of them that allow us to draw the early bridges in this field. Now, they propose a direct test of whether or not consciousness has sentient ability by building a quantum computer and connecting it to the brain to the proper biological substrate and seeing if this accompanies an extended sentient space. And they're planning on looking for the actual biological material with quantum channel tests that they're actively doing right now in Santa Barbara. Now, what are these quantum correlates of sentience that they're interested in and that I'm interested in as well? So these are the 5 main ones put in their absolute simplest forms I can imagine, where we'll start with the quantum state space, where in quantum mechanics, we have these really interesting Hilbert spaces that emerge, where we have n qubits, we require 2 to the n dimensions to fully describe them. Now, there's lots of objects in mathematics that have high-dimensional spaces required to describe them. You could imagine a very complicated stress tensor on a bridge having a high-dimensional space that describes it. But no one really proposes that those dimensions are real or should accompany new dimensions that are appearing in the world. In quantum mechanics, these dimensions do seem to be real. And this comes down to the PBR theorem, which is a recent result in quantum information theory put forth in 2012 that has seen quite strong support since then and is yet to be disproved in any meaningful way. Now, what the PBR theorem states is that in order for quantum mechanics to act the way that we know it does, and to produce entanglement in the world, and for the Bell inequalities to be broken in experiment, the wave function must be ontologically real. We can correlate this to something in our reality that needs to fit these extended dimensional spaces of quantum mechanics. And when we're looking for new dimensions, new dimensional spaces, many orthogonal dimensional vectors to look along, the qualia appear as a very attractive substrate to fill these. Now, the qualia, which are the we'll take a step back and just sort of define sentience at the top right there, where what is sentient is that feels or is capable of feeling, having the power or function of sensation or perception by the senses, which is a very old definition of sentience that has held since 1632 in the Oxford English Dictionary, and is prior to consciousness, where consciousness is sometimes correlated to sentience, and someone like Hameroff would use them in essentially the same way, where what is conscious for him is what is here is sentient. For many other people, though, consciousness requires higher processing abilities. So I've chosen to use the word sentience here just to sort of define the very base level. This doesn't require awareness or something like passing the mirror test, which would be a very developed form of consciousness that we see only in certain animals. This form of sentience is as base as it comes. It could be purely the experience of red. So if you could imagine a pure experience of red, and something might not even know that it's experiencing such a qualia, but this is just the very pure, raw state of consciousness, the very base level. Now, these qualia exist within a large number of orthogonal dimensions, and this is from studying our own internal state. Several of these dimensions could be considered the color space that we see in for vision, or the sound space that we hear in, or the emotional space that we feel in. And all of these spaces are very orthogonal to each other. We can't measure our own experience of a certain tone, a certain musical tone, by a purely color qualia. So these are purely orthogonal dimensions that do seem to exist, as at least I experience them, and many others report experiencing them. Now, another element of quantum mechanics that we see is coming from Bell's theorem, where quantum information is known to be non-local. And this is required in order to violate Bell's inequalities, which have been experimentally tested and led to Nobel Prizes at this point. And the qualia themselves are experienced simultaneously, which is very confusing to many philosophers. This is known as a binding problem of consciousness. How are all these individual qualitative elements bound together? And this creates a sort of non-locality in the qualia, where we experience all of them in a singular bound moment. We don't experience them in a chain of individual qualia. It's one large super qualia that forms our internal world. Now, we've also taken from quantum information theory the no-cloning theorem, where it's impossible to create an identical copy of an unknown quantum state. This is similar to how it's impossible to know a foreign qualitative experience, which is known as inner privacy, and it's been very deeply explored by the philosophers. And you can see the essay, What Is It Like to Be a Bat?, which famously explores this question, how we can never truly know what it's like to be inside of the experience of a bat, in a similar way that we can't directly copy a quantum state to create an identical form of it. And the fifth identity that we're looking at here is Holevo's theorem, where the maximum accessible information from n qubits is n classical bits. And this has been compared to the human experience of trying to convey our own sentient experience to another person. Now, if I wanted to tell you what I'm seeing, I can't directly port to you my entire sentient experience of the world with all of its qualitative aspects. I need to condense it into a classical string of information, which then I can convey to you in many different forms. Right now, I'm using my words to convey them to you in a classical string of information, but a bumblebee might use a dance to convey it into a mechanical classical form of information to communicate. Now, going into these papers a bit deeper, beyond this very surface level look, we can briefly look at the PBR theorem and the results that they get, where they're seeing that quantum states, which are the key mathematical objects in quantum theory, are very undecided to what they really represent. Many people say that they correspond to reality, while others say they just represent knowledge or an information about some aspect of reality. In the PBR theorem, with the assumptions that they make, they show that any model in which a quantum state represents mere information about an underlying physical state of the system, and in which systems that are prepared independently of independent physical states must make predictions that contradict those of quantum theory. Now, this does require some assumptions still, and further work afterwards have eliminated some of these assumptions while holding the core value of the theorem, which has made it even stronger since then. These authors finished the paper by saying, on a related but more abstract note, the quantum state has a striking property that the number of real parameters needed to specify it is exponential in the number of systems n. This is to be expected. The quantum state represents information, but to the authors, it was very surprising if it has a direct image in reality. So these are these rapidly expanding dimensional states that we were just talking about that seem to correlate quite well with the many dimensional states that we qualitatively experience in our inner experience. Bell's theorem, which is quite famous, we can look at a conclusion from it where, moreover, a hidden variable interpretation of elementary quantum theory has been explicitly constructed, where Bell is directly talking about Bohm's theorem, Bohm's pilowave mechanics here, which has a grossly non-local structure in his own words. And this grossly non-local structure, which was initially one of the most disliked aspects of Bohm's pilot-wave mechanics, is characteristic of all quantum theory nowadays, post-Bell's theorem. So this is just saying, essentially, that certain physical variables must be non-local to agree with the predictions of quantum mechanics, even when position and velocity remain as local variables. So these non-local variables may, in fact, be the qualitative states. But we still need more experiment that tries to dice down and actually prove this to be. Now, briefly looking more into the no-cloning theorem, the authors here asked, but is it possible by this or any other process to amplify a quantum state, that is, to produce several copies of a quantum system, each having the same state as the original? This was not possible. That the linearity of quantum mechanics forbids such replication. This conclusion holds for all quantum systems, which, as a sentient being that may be correlated to a quantum state, makes me feel a bit more comfortable that no one can directly copy my mind and create a pure clone of me to run around the world somewhere. Now, why am I calling this sentient wave mechanics? Right now we have sentient quantum mechanics of some kind, but as Bell alluded to, how any grossly non-local version of quantum mechanics is roughly equivalent to Bohm's and de Broglie's original pilot wave mechanics, we can start to ask, how do these qualia really interact with the material world? And that's where the growing world of modern pilot wave mechanics starts to become interesting and helpful to us. So what we're looking at here are real physical experiments of particle wave dynamics. These were originally a spurious accident where a physicist flicked a drop of oil using a toothpick onto a vibrating bath of oil and noticed that his little droplet began to walk around with an associated wave field next to it. In the top left, we can see an individual droplet bouncing and generating a wave field. On the right, we can see two droplets walking away from each other with wave fields. And at the bottom left, we start to see what happens when the wave fields interact. So these particles are actually still bouncing on the bottom left. You're just using a strobe light to make them seem like they're standing still vertically. And the wave fields allow them to interact while retaining their particle-like behavior in the very center. We can get crystals and many higher order structures. When these physicists began to experiment further with these systems, they were able to get features such as a single slit diffraction and double slit diffraction, which, according to Richard Feynman, is a phenomenon that is impossible absolutely impossible to explain in any classical way, which has in it, the heart of quantum mechanics, which is, in his words, the only mystery of quantum mechanics. Now, what you're looking at right here is a classical demonstration of double-slit diffraction, whereby our particles, dressed in the wave that's associated with them that they are driving, the particle is able to go through one of the slits, while the associated wave field propagates through both slits, allowing this wave field to interfere with itself and give us wave-like statistics with a single particle moving through them. They were also able to recreate particle-Noel dynamics, whereby the particle, as it spends its time exploring this potential well, which is just a different height under the oil bath. So we have our oil on top of a three-dimensional bath, and the particle explores this, and after it explores it for a long period of time, we get the same statistical behavior as we see for a single electron in a potential well. Now, this suggests that for the three-dimensional classical walking droplets, where we have this statistical behavior, which is based on an exposed variable theory, that there could be a hidden variable theory of quantum mechanics very similar to those originally proposed by Bohm and de Broglie. They were also able to recreate the phenomenon of tunneling with the same statistics as traditional quantum mechanical tunneling based on barrier width, as well as showing mode superpositions between different states. Now, they were able to go on to show particle voids annihilating, similar to an antiparticle and a particle coming together. Here they have a bubble and a droplet that are just about to annihilate. Boom. And what's very interesting, that it leaves behind the wave field that contains the memory of where these particles walked before in their interactions. They're able to create spin lattices, Friedel oscillations that we see in scanning tunneling microscopy. Very famous, the quantum bomb tests as well as the phenomenon of super radiance. And all of these, if you look in the bottom right of the slide, you'll see the citations, the papers, all very well published papers. These authors have been making great waves in theoretical quantum mechanics coming forward. Here we can also see the quantized orbits, very reminiscent of our molecular orbitals from the hydrogen probability density, where if we look at these molecular orbitals and then we look at the quantized orbits that these walkers show in different sized wells, we can see a strong similarity between them. And the picture of the subatomic world starts to become a bit clearer. Now, we also see similar results in photon trajectories when we try to look at the actual path that an individual photon will take through a sample, which we cannot do for an individual photon, as taking a full measurement of it will stop the photon from propagating. But in the area of weak measurement, we can weakly measure many photons and start to reconstruct the average trajectories they may be taking. And these trajectories, in the words of the authors here, as published in Science, recreate the trajectories of the Bohm-de Broglie interpretation of quantum mechanics. Now, this isn't entirely true, as Bohm and de Broglie actually had two different theories that are often conflated, and they all fall under a family of pilot wave theories, of which there are now many. Quite interestingly, Bohm's pilot wave theory was self-stated to be wrong. It was only a temporary theory where he was trying to show that such a theory was possible to recreate the statistics of known quantum mechanics. But he thought it should be further developed in order to have a rational underpinning. This has been done by John Bush, whose experiments we've been seeing, John Bush and Yves Couder. And the mathematics have been developed since this is de Broglie's. And with Bush's mathematics, we can start to see these hydrodynamic quantum field theories, which he leaves us with a single question. So what is the matter wave field in quantum mechanics? we start to ask, where are these waves coming from that might be interacting with the particles that make up matter? Now, if we view these waves as being the possible quantum correlates of sentience, where qualia start to fill in these non-local variables in our pilot wave theories. So the qualia are the actual non-local variables. We can see how the qualitative aspects of reality might interact with the physical matter aspects of reality. So this is just a brief motivation of why we start to correlate quantum mechanics with consciousness. So do we have any questions here before we start to move into the cytoskeletal aspects?
Yeah, I have one specific question and then one general question. We'll start with a specific question. Would you mind explaining a little bit of the results of superradiance from that one slide, just briefly, because I find it personally interesting. I think the audience would too. And yeah, I'll let you go from there.
Sure. So superradiance is a phenomenon that is now getting a bit more attention in the consciousness crowd due to recent experiments showing superradiance in microtubules and cytoskeletal structures. This is not the first area that we've seen superradiance in biology. Superradiance is well known to play a role in photosynthesis, and that research has been going on for over 2 decades now. But superradiance in general is where we have multiple emitters emitting coherently in phase. And we get much larger emissions that scale with the number of scale exponentially with the number of emitters. So right here, what they're looking at is we have 2 individual emitters that are driven by this coherent wave field in the bath around them, allowing these wells that would not normally be able to emit a droplet on their own, when they're placed near each other and coherently coupled to each other through this classical wave field, we get this large emission of a droplet coming out of both of them.
Okay, yeah, thanks, Daniel. So yeah, I'll let you continue. I'll save the general question for the end.
Great. Okay, now we're moving a bit more into cytoskeletal work, which is really where I've spent most of my time. A lot of that foundation of quantum mechanics has just been a recent excursion that I've taken trying to motivate the quantum consciousness work. But more of my experience comes down to the hard quantum biology. What experiments let us see quantum effects in biology, and where do we see them? So the cytoskeleton is one place that's been spoken about for a long time due to the work of Penrose and Hameroff. So let's start by introducing the cytoskeleton. Now, the specific aspect of the cytoskeleton that many are looking for quantum effects are the microtubules, which are nanoscale tubes that form the skeleton inside of our cells. So the cytoskeleton is our cellular skeleton. On the left, we can see a model of one of these microtubules where they're having about a 24 nanometer diameter, and they can have very large aspect ratios. So while they have this nanometer diameter, they can be millimeters long, potentially. In say, a neural axon, you might get a millimeter long, which leads us to an aspect ratio that's similar to a carbon nanotube. Now, they form from these tubulin bricks, which are built out of alpha and beta tubulin units, 2 closely related units that come together to form our tubulin block, which you can see here in the purple and blue pieces coming together. And then they assemble in a spiral fashion to form sort of a spiral brick chimney, you can think about it. They are most famous from their roles in cytokinesis, which we're seeing in that central video right there, where we have the microtubule spindle that assembles, reaching forward, organizing our DNA, and then pulling them apart to separate into our 2 mitotically dividing cells. This process, in animals at least, is coordinated by the centrosome, which has 2 centrioles inside of it, which is what we're seeing at the bottom there. Now, the centriole is a barrel-shaped organelle composed of microtubules and many fibrillar proteins, as well as cytoskeletal motors that hold it together. And we have two of these barrels at a 90-degree orthogonal angles, as we can see them below. And these structures are incredibly precise geometric architectures that have been part of the animal lineage since the last eukaryotic common ancestor. So we find them all across the eukaryotic world, where they act as the central points of our cytoskeleton. So every microtubule in our cell is typically nucleated off of these centrioles, not directly on their surface, but slightly outside of them. In the centrosomal matrix that forms around our two centrioles, our microtubules nucleate there, grow outwards, and either stay nucleated there and attached, as in the mitotic spindle, or they can drift off into other locations in the cell for various purposes. Oftentimes, these purposes are for shipping things around the cell, shuttling various goods or vesicles or mitochondria using the motor proteins that are able to walk along our microtubule surface and move things around the cell. On the right, we can see some characteristic cytoskeletons of different cells, where in the top we're looking at a neural growth cone with our microtubules going through the axon to the top of the growth cone and then shifting over to actin filaments around them, where actin is our other major cytoskeletal protein. Instead of a rigid tube like the microtubule, it's a twisted fiber protein. And you can sort of think of them like the beams and ropes inside of our cell. In a migrating cell, as we see below, our keratocyte, we can see a more diffuse microtubule network centered around our nucleus, with our centrioles inside the centrosome, sitting in a little pocket right next to the nucleus, and then another diffuse cytoskeletal network in our fibroblast as well. Now, as microtubules and centrioles have been around since the last eukaryotic common ancestor, we see them in many different organisms. On the very top left there, we're seeing a little illustration showing which cytoskeletal proteins are present along various evolutionary stages, where in the bacteria, we don't have any of our alpha tubulin, beta tubulin that makes up the microtubule, or the main actin proteins that make up our fibrillar proteins of our cytoskeleton, or any of the motors. So none of these standard cytoskeletal elements in the eukaryote are present in the bacteria yet. By the time we get to the early eukaryotes, such as the yeast, our full complement of actin proteins are in, our alpha beta tubulin are here, and all of our motors in the cytoskeleton are part of it as well. Now, for the more complicated single-cell eukaryotes, such as the ciliates, this is where we get our full complement of cytoskeletal proteins that are present in animals, such as us, or the higher eukaryotes in general. And below, on the bottom left, you can see a filamentous fungi with a microtubule network inside of it. And on the very right, we see our mammalian neuron with the microtubules in blue and the actin in red. While centrally, we see a ciliate, which are essentially single-celled animals in the amount of complexity that they pack into an individual cell. All the little hairs we see coming off the ciliate there are the cilia that they derive their name from. And there's many different forms of ciliate, but Tetrahymena thermophila, as we see here, is one of the most studied. And below that, we see a depiction of an average cilium with cross-sections along its various tip, mid, and base, and little diagrammatics of what's inside of it. So you can see the central microtubule cores, as well as our radial spokes in green and linkers attaching it firmly to its membrane. And these just sort of show us how these early single-celled eukaryotes are packed full of microtubules, have very developed cytoskeletons, And this characteristic arrangement of cilia you're seeing in tetrahymena is passed from one to the other, showing the exact same arrangement on all of them in a similar fashion to how animals pass on our arrangement of organs and subcellular or suborganal structure within our cells. The ciliates pass on their arrangement of organelles, and their internal structure inside of the ciliate is very highly organized as well. Similar to how our organs each have a position in our body, each of their organelles is a very specified location inside of their body. Now, cilia are quite capable organelles. They're conserved from our single-celled ancestors, and actually from the last eukaryotic common ancestor, where they first showed up. How they got into the last eukaryotic common ancestor is a bit of a mystery still that we might touch on later, but the first cilia that we ever really see show up fully formed. They nucleate from centrioles. So on the bottom left, we can see our centrosomal centrioles migrating, merging with a vesicle, then merge with our cell membrane, and allows the cilia to grow essentially as an extension of our centriole. Now, cilia have been evolved by animals towards the most capabilities, where in a human, you have many different kinds of specialized cilia, which are the sensory cilia. And the rod and cone cells in your eye are an example of a sensory cilia specialized for phototransduction, getting different kinds of photons and allowing us to have vision. The hair cells that will give us hearing in our ear are templated by kinociliums, which are specially tuned for mechanical frequencies. In your olfactory senses, in order to have smell, we have chemosensory specialized cilia, as well as in our kidneys to beat fluids and help absorb different nutrients. We have cilia there. And then every single one of your cells will also express a primary cilium that has a whole host of sensory abilities around it. These include mechanical, chemical, optical, magnetic, gravitational, osmotic, which is the state of the internal state of the cell, and thermal sensing. And they are key to cellular differentiation and the development of an organism as a whole, sort of integrating all the cells and letting them know where they are in their environment so they can differentiate in the correct direction. Now, centrioles and cilia are quite small nanoscale features that have only recently come into molecular focus. These presentations that we're seeing right here came out earlier this decade, where we can see the molecular architecture of the centriole and the molecular architecture of our ciliary structures. So looking at the centriole, you can see this very geometric architecture that has been conserved since the last eukaryotic common ancestor, which helps a lot for studying it, since we can just as well look at a human centriole, or we can look at an algae's centriole, and we'll see the same geometrical architecture. It's not entirely clear why this architecture has been so highly conserved. And nowadays, we are seeing that there are slight deviations that can appear in some divergent eukaryotes. However, in order to form these cilia that are so foundational to animal sensation, we do need this very precise geometry in order to form any cilia that we've seen so far. And on this slide, towards the left, you're seeing a typical motile cilium, which has microtubules running down the center of it as well, with motors connecting them, allowing it to beat fluids, or for an algae to swim through its viscous environment, while the primary cilia does not have these centralized microtubules, and they can't produce any motion on their own. They're purely for sensory purposes. Now, cilia have been quite interesting for the world of quantum biology, as many of the quantum biological hypotheses that have been raised, or the quantum biology experiments that have confirmed various quantum effects in living organisms, but specifically the eukaryote, exist within the ciliary microenvironment. One of the most famous of these that we'll start with is cryptochrome, where radical pair entanglement allows birds to track the magnetic field, whereby how this operates is an ultraviolet photon comes in, hits our cryptochrome protein, excites an electron, splits our pair. Our electron traverses a chiral bridge of tryptophan to get to the other side of our protein. And during this process, it remains entangled. And very weak magnetic fields cause these entangled electron pairs to slightly precess, leading to different downstream chemical products, allowing the bird to tell which direction it's facing in the magnetic field, which can be very helpful while it's migrating across an ocean, say, in the middle of the night. And it has no visual cues. It has no stars that it could follow if there's a cloudy night, but it can still stay exactly on track due to this quantum-enhanced magnetic sensor. Rhodopsin has also been known for the past 2 decades at least to utilize vibrational coherence in its function, which is also stored in the ciliary microenvironment of the rod cell. And the ion channels that are present in its membrane, there have been several proposals put forward that there may be a form of coherence going on, or tunneling in order to allow the rapid transit through ion channels. The experiments in this region are less well developed. I haven't seen anything directly proving it wrong yet, but it seems to have been difficult to actually show this coherence or tunneling effect in action. What is definitely more well proven is the electron tunneling chains in mitochondria, which can traverse, I believe, almost a nanometer. They can tunnel electrons. There's also proton tunneling that's taking place inside of the mitochondria, allowing them to bypass classical energy barriers and give us all the energy that we need to power our body. Now, what we're mainly going to focus on from here. Oh, quick question.
Yeah, I just wanted to riff on one thing about the mitochondria. In vegetables and plants, photosynthetic systems, it's also been demonstrated experimentally and theoretically that there is quantum tunneling of electrons as they go from the surface of the photons, rather, as they go from the surface of the leaf into the energy systems of the plant, the chloroplast. So, and that's all to say that with these 3 examples you've given and the photosynthetic example, quantum effects in biology are a well-known and studied and quantified phenomenon, which is awesome. So I just wanted to riff on that a little bit.
Yeah. So all of those quantum effects I was talking about previously are the eukaryotic ones in animals specifically. Now, in plants, we definitely have way stronger evidence of quantum biology that at this point is firmly grounded and accepted and beyond accepted, that it's now turned into bioinspired technologies based off of these quantum biological systems. Where on the left, we're seeing these photosynthetic antennas that utilize quantum coherence in order to direct and funnel our excitations, where they sort of act like you think of a benzene molecule, where you get your resonant electrons across the entire benzene. They act like a supermolecule, where you get these coherent resonances across the entire antenna structure. And this has been developed into these light harvesting nanotubes, originally by Munji Bawendi of quantum dot fames, but are now being developed further at Justin Karam's lab at UCLA, who's right down the hall from me. So I've had the opportunity to talk to him in depth about these bioinspired quantum architectures that utilize these large-scale coherences across the entire supramolecular structure in order to hyper-efficiently harvest photons from the environment. Now, the actual tubes that were developed following these, coming out of these bioinspired photosynthetic systems, have much more in relation to microtubules in their structure than they do to the original photosynthetic antennas, where they figured that in order to enhance their effects even further, they decided to create these one-dimensional nanotubes through their intelligent design of their supramolecular systems that get even greater enhancements when they form these sort of bundled structures that you're seeing in the blue box on the right, where these nanotubes that have very similar dimensions to microtubules, as well as using the same optically active indole group as the tryptophan that gives microtubules their supposed quantum biological properties. So these intelligently designed supramolecular structures that showcase really great superradiant properties have a lot in common with the biological microtubules that we'll be talking about going forward. Now, briefly motivating this work as well that we've been talking about, centrioles appear to have a central role in cancer, which can sort of make sense, and it was originally hypothesized before the centriole was even directly visualized at all. The early biologists, early microscopists looking at cells knew that the centrosomes would form on either side of the cell and pull our DNA apart. So they hypothesized that in cancerous tumors, we might have extra centrosomes forming, creating additional points trying to pull DNA towards them. And in more recent studies, it turned out they were correct, that in cancerous cells, we have supernumerary centrosomes where we just have lots and lots of different cell center points that tear our DNA in all sorts of directions. And so if we can start to understand the quantum biological properties of the centriole and cytoskeleton, we may have new ways of interacting with cancerous tumors. Now, onto the actual quantum biological effects. The primary paper that supports this is a recent publication in the Journal of Physical Chemistry coming out of Philip Currien's lab, the quantum biology lab at Howard Institute, where they did a dual simulation and experimental work looking at mega networks of tryptophan in microtubules, centrioles, cilia, and neuronal axons, where neuronal axons are just typically are formed by hexagonal lattice bundles of microtubules that form the central component of the axon. And in their simulations, which we're seeing on the right, as the number of tryptophans increase, we see this first jump up this step during the formation of our first ring of tubulin, and our second growth here is going on as we start to extend our microtubule. And we see these slight increases in quantum yield, which are essentially for the photons coming in, how many of them do we capture and utilize. So in their simulations, we see these increases in quantum yield, which could be due to many different things in theory. But what they believe is happening is that we're getting this collective interaction between all of these individual tryptophans that allow them to act very similar to the photosynthetic antennas that use superradiant states to collectively interact with incoming photons and capture them in this supramolecular state. So the supporting simulations, they demonstrate the emergence of superradiance as well as subradiance, which is the out of phase sort of mirror to superradiance, where superradiance is an in phase phenomenon, where all of our individual molecules, which are tryptophans here, oscillate in phase to collect a photon, all sort of oscillating together, absorbing the photon. Subradiance is when all of them oscillate out of phase, and we get the same distributed quantum coherence across all of our different molecules. But instead of an ultra-fast phenomena, we get an ultra-slow phenomena, where the energy sort of oscillates across all of our molecules. And anytime we have superradiance, we also have the potential of subradiance. It's just much harder to show subradiance experimentally, as subradiance is what's known as a dark phenomena, as when the photons are absorbed into these subradiant states and are oscillating, they're trapped. So it's a non-emitting state. And typically the subradiant states, when they're just slowly oscillating the energy around, will release it through molecular vibrations, and the energy will just sort of leak out into the environment. Where in the superradiant states, we get a bright burst of light that we can record and experimentally show it to be present. Now, in their simulations of these quantum optical superradiant effects, they appear to be very robust under disorder, even disorder significantly greater than what would be expected at body temperature. Now, there are other systems that show superradiant behavior. The photosynthetic antenna was the one we'd already spoken about. The artificial nanotubes that were designed based off our biological systems are another system that show distributed superradiance, as well as much larger photosynthetic systems as well. So these superradiant effects aren't contained to an individual little barrel-shaped chromophore. They sort of extend over the entire antenna array, where we start to get entangling factors and quantum coherences across these entire photosynthetic antennas. And the actual superradiant enhancement seems to scale with our system size as well. So as we start to create these larger biological systems, we get even larger superradiant enhancements. Now, this likely doesn't hold for any arrangement into a larger and larger system, but these biological systems have evolved seemingly to take advantage of this. In another bioinspired, semi-bio, semi-synthetic system, these are viral capsids that have been tagged with fluorophores, and they have also been experimentally shown to demonstrate superradiant effects, where they have this quantum coherent collective emission. And what was quite interesting is that viral capsids are a very well ordered structure. They're these perfect little icosahedrons. And the actual ordering, this icosahedral order, appeared to be very important for these effects to show up. That if they introduced significant disorder onto their surface or broke these capsids open a little bit, they would no longer get these superradiant quantum coherent collective emission effects. Now, this is something that I found fairly interesting as well, that many of these, on the top, we're seeing 3 different photosynthetic antennas from low light bacteria. So photosynthetic bacteria that typically exist in the bottom of the ocean, where they're still trying to get light, but they're in a very light deficient environment. So they'll typically be receiving a countable number of photons per second. So somewhere in the range of 10 photons per second. So for them, every photon is essential for their lives. And they all seem to focus on these 9-fold antenna symmetries. Now, in purely simulated systems, we can see these huge increases on these plots in the bottom, increases on the absorption of coherent and incoherent light at this 9-fold symmetry when our individual emitters are placed at a sub-wavelength distances. So that just means that the distance between each of our emitters here is significantly smaller than the light that they're interacting with, which is true for both the photosynthetic antennas that we're seeing above, as well as the centrioles and cilia that we're seeing below, which are hypothesized to interact with ultraviolet light. Here we can see the actual distribution of radiance states along the photosynthetic antennas to sort of get a mental picture of what these look like, where the superradiance state here is what's used to focus the incoming light, absorb it, and then focus it onto the photosynthetic reaction center. So you can see in this, in the most superradiant state that these photosynthetic antenna systems have, they focus almost all of the absorbed energy directly onto our reaction center, which is what allows us to have these hyper-efficient reactions in photosynthesis, while the subradiant state is sort of distributed all through the cytoplasm around it and is less likely to be populated in the photosynthetic systems, which are trying to funnel the energy into our reaction center, but is potentially populated in our cytoskeletal systems where we don't know of any reaction center that they would be focusing this energy into. Now, what a more radical proposal, what these cytoskeletal systems may be doing with these quantum optical properties, is using them for some form of quantum processing. Now, superradiance and subradiance have been proposed for optical quantum processing, as they do maintain coherences. And superradiance can be used to sort of transfer our optical information onto an antenna array, which can then be stored in these long-lasting subradiant states that we have. So on the left, we're seeing artificial simulations of using superradiant effects between different arrays as quantum processors, where on the right, we're seeing our centrioles, which may be transferring optical quantum information between each other, or more distantly, between individual centrosomes across our microtubule bridges during mitosis, or potentially further along something like a neuronal axon. Now, a bit more about centrioles, where they actually come from. In animals, these centrioles come with the nucleus in the spermatozoa, where we'll have our two centrioles. One of them will be stored right next to the nucleus, and the other one will be forming the base of the ciliary tail. And these two merge with the egg, who has dissolved her centrioles. She'll actually dissolve her two and turn them into a pool of free tubulin dimers. And the two sperm centrioles will come in and then be matured by this maternal pool to form the first two centrioles of any animal. And then from there, every further centriole will be directly duplicated off of the surface of these of the previous ones. So the first mitotic event, it will be duplicated off the surface of the sperm's two centrioles, and then from there we'll just continue duplicating, directly passing this down, potentially passing down entanglement between all of these states. And so in an animal like ourselves, where we have trillions of centrioles, they will have directly duplicated from these initial two and are potentially remaining entangled throughout this entire process, creating a distributed reservoirs, or a set of distributed reservoirs in each one of our cells that have some of this initial quantum state from our initial centrioles that sort of allow our entire body to have a single quantum state diffusely distributed throughout the body that may be correlated to our internal sentient state that we have, where we have many different qualitative dimensions, lots of detail inside of them, huge amounts of emotional space as well, that sort of may be stored within these cytoskeletal elements. Now, I think this is probably a good time to pause briefly if we have any questions so far, anything we want to go more in depth in.
Yeah, I just want to say I just wanted to say first of all, Daniel, these slides are absolutely beautiful. Um,
Thank you.
one specific question I had back when you were describing the evolution of the cytoskeleton, touched on prokaryotes up into yeast and multicellular organisms. You briefly mentioned that we don't know much about how life made the leap, so to speak, from relatively simple cilia-free cytoskeletal structures to our modern, more robust cilia and cytoskeleton structures. You said we might speak a little bit more about that. If that's forthcoming, I'll wait, but if not, maybe you could touch on that a little bit. And then the second related question would be, in terms of prokaryotes and bacteria, they do have tubulin-like proteins like the FSZ, if I remember correctly. And I've shared this paper with you before about how they don't make the typical microtubule structures, but they do have these 3 or 4 filament tubes that they do generate. I was wondering if you knew any related work on cilia and perhaps quantum effects in those older structures and how that ties into the evolutionary history of these.
Yeah, definitely. So here I'm pulling up a one of the proposed potential origins of the centriole and the eukaryotic cytoskeleton. This is the viral hypothesis. And yes, if we go back to that initial slide, FTSZ is a very reduced and earlier form of tubulin that in bacteria does form helical and much smaller tubular filaments with like 3, 4, or 5 filaments instead of the traditional 13 that we see in the eukaryotic microtubule. So the bacteria does have FDSE, it has some ability to form these tubular filaments. They don't exactly act like the cytoskeleton by moving around or interacting with the genetic material in the same way. From what I know, it's not entirely clear what they were doing at the time. But if we go back to the viral evolution, where we can start the simplest viruses are just helical proteins, which are almost similar to these early FDSZ-like tubes as well, where many different protein bodies that form self-associations. If this self-association has some sort of slight angle to it, they form the ability to create helices. So lots of different early viruses form different helical bodies in a similar way to these FDSE proteins forming early helical tubes. And then next we had the icosahedral viruses start to show up. From quite early on in the viral, even pre-DNA, RNA world, where these use quasi-crystalline templating, where we have the same protein at almost every single point along this virus, and they're able to form these true icosahedral shells, which is your icosahedron is the classic D20 for anyone who's played D&D or dice games. And then in the complex viruses, we start to get interactions between these different parts as the viral world progresses. Where here we see our helical virus sort of associating with an icosahedral virus and forming this complex virus structure. And at some point, these more complex viruses or larger icosahedral viruses were infecting prokaryotes such as DNA or such as bacteria. And they started to form endosymbiotic relationships with them, where by the time we get to something like a giant virus, which they're significantly larger, we can see it's almost 500 nanometers across, so significantly larger than the tens of nanometers of a typical viral particle, how these much larger creatures are starting to form is that the virus, which was initially a pure icosahedral virus, took over some bacteria and then almost wrapped itself in the bacterial membrane and took a lot of the bacterial proteins with it, including the FTSZ microtubules. So a lot of these giant viruses now having this bacterial membrane, having the early cytoskeletal proteins that appeared in our bacteria, start to do more complicated things with them, including, very interestingly, the ability to form a mitotic spindle and creating a nucleus in the center of the prokaryotes that they are able to infect, or the eukaryotic cells nowadays that these giant viruses are able to infect. And this is actually believed to be the origin of the eukaryotic nucleus as well, where these viruses coming in form a nuclear viral factory where they produce lots of themselves. And they center this viral factory inside of the cell using a microtubule spindle, which has led these authors to suggest that both of these organelles may have showed up at the same time. Some centriolar seeming giant virus that comes in, creates a nucleus with a spindle, and at some point, after infecting the perfect archaea, was able to form the last eukaryotic common ancestor in this way.
Yeah, it's just very poetic to imagine that the light of consciousness might have passed through the loving relationship between a virus and its bacterial host. I mean, it's very psychedelic to think about.
Yeah. I've always wondered what an endosymbiotic event might feel like. Or we see similar things going on in ciliate reproduction as well. How some ciliates, they can either just divide on their own to produce, or they can come together with another ciliate, merge their membranes, come together, form a single organism, swap DNA, and then go off. This is, to me, one of the most romantic events in the biological world. Now, here's just a very pretty video and illustration coming out just last year of the assembly of a centriole. Doesn't tell us much about the quantum mechanical properties. It's just showing us how these objects actually form in the cell. We can see the initial templating architectures on the bottom and the microtubules growing outwards. Now, this would be coming off of, at a 90-degree angle, coming off of the mother centriole as well as it grows outwards. Something else of interest to us right now, when we look at centrioles, they don't have perfectly 90-degree walls. They're slightly curved inwards and create what appears to be quite similar to a confocal cavity. And this cavity in water, it's the primary excitation peak of this cavity is the same as the wavelength that tryptophan are excited at, which we haven't been able to show that this cavity effect is happening, but it is suggested that there may be some sort of cavity effect with the ultraviolet light happening inside of centrioles as well. Now, something that would be confusing is how is the walls of the centrioles acting as the mirrors of a cavity, where typically for forming a nanoscale cavity, we'll have some highly polished gold. However, in subradiant mirrors, which is a recently introduced technology, when we take multiple emitting species, put them in a regular array, and have some sort of subradiant interaction between them, they can act as a highly efficient mirror. And for the actual spacing of the centriole, we'd be coming in right around here. So we'd be, there may actually be some real mirroring effect for this ultraviolet light, not due to the sort of bulk properties like a gold or metallic mirror we would see, but more due to these subradiant nanoscale interactions going on. Now, this is just a very good illustration of, before I was talking about the structure of the neuronal axon, having these hexagonal lattices of microtubules. So you can see this dense hexagonal lattice of microtubules here. Here's a SEM image with everything else sort of stripped out, allowing us to see the individual microtubules connections in between them. And then on the right, a cross-section of this axon showing the very well-ordered hexagonal bundles, which are very reminiscent of the synthetically created light harvesting nanotubes that also show their greatest subradiant ability. Oh, I don't have the cross-section here, but it's a very similar hexagonally packed bundle as well going on in these synthetic light harvesting nanotubes. Now back to where we were in the presentation. This is a very interesting open question that I don't have an answer to going on. But the centriole appears to act as a set of eyes in the cell. This is coming out of Albrecht Büller's work on the centriole, where he was able to show that cells can locate a pulsed infrared source and sort of reach out, a little Filipota, reach out an arm to check out what's going on at this pulsed infrared source. And in his belief, he thinks this light, this pulsed infrared will be similar to the light being emitted by a cell body. And it's how cells sort of can see each other, interact with each other. And he was able to show this is not due to heating, as this effect persisted when there was a wall placed between them, so no fluid, no temperature gradients allowed to introduce this, that the cells are able to locate the light source, reach out an extension towards the light source, and they're able to continue doing this except for when the centrioles are removed. So every other organelle could be removed aside from the centrioles, and the cell would still be capable of locating and reaching out. And based on Albrecht Büller's hypothesis, the centrioles are a near optimal arrangement in order to act as the center point of a 3D coordinate system in order to locate where these signals could be coming from.
That is.
Although, to my knowledge, it's not exactly clear where they're absorbing this infrared light, as the tryptophan absorbs into ultraviolet. It's potentially a cavity mode. That's my only hypothesis at the moment, but we've yet to do anything to test that. Oh, this is a brief slide on why ciliates are a great model system instead of working with animals. Ciliates have a similar sensory integration where they have chemical, optical, gravitational sensing that they're able to integrate all that information together to create decisions. They have left-right asymmetry, hierarchical body planning, and regenerative abilities to make them quite nice to test with, as well as having genetic, cytoplasmic, membrane, and cytoskeletal inheritances from the last eukaryotic common ancestor. So we're all sort of on the same branch from an individual organism that once lived. Now this is some interesting work showing a bit of almost scale-free dynamics where around an individual centriole, where microtubules are actually nucleated from, they're called nucleating bodies or centriolar satellites. These are little bodies that sort of float around the centriole. And if we look at the arrangements of the individual bodies, as in C or D, which is right around, this is within the centrosome, and then we zoom all the way out to look at the structure of the cell, we can see some clear correlation between these architectures. So the actual nucleating points right around the centriole show up again in the absolute structure of the cell on its largest scale. This work by McNiven and Porter, where Porter was actually one of the most important cell biologists of the last century, I believe. There's some really interesting writing about him. But he was good friends with, good friends with Solomon, who did very interesting work on neurites and their morphological memory. So what we're looking at here on the left is 3 neurites, which are given a drug that disassembles your microtubules, and it disassembles all the cytoplasmic microtubules. Now, centrioles are much harder to disassemble, so in these preparations, the centriole would actually stay together, but every other microtubule would just turn into the tubulin bricks and sort of be dispersed throughout the cytoplasm. And after disassembling them, waiting a significant period of time, we can see the cells start to reassemble themselves, and at the very end, they form very similar geometries to what they were before the disassembly event, which shows that there's this potential of them having a memory or a desire to go into a certain morphology. However, there could be some sort of small traces on the glass. So his work also looked into sister neurites, where two cells are allowed to divide and then grow from there. And on the right, you can see in each one of these individual boxes, we can see a set of sister neurites that form mirror images of each other in the way that they develop. Where this is definitely not following any sort of chemical traces on the glass, but it's showing that they have some sort of predetermined goal for their morphology as they're growing outwards.
Yeah, it looks like instinct to have a
Potentially
macrobiological
an instinct on the cellular level. Now, this is from Anirban Bandyopadhyay's work, where he's created a radio frequency STM, where he's able to excite microtubules through the tip at various radio frequency and then image them using scanning tunneling microscopy, so tunneling electrons off the surface. And we start to see these topological features appearing in several of the images on the bottom right, where on the first image, without any radio frequency excitation, we just see the single microtubule as a large object, and we just get sort of a topographical view of it. Where on the bottom we see, especially in these two images, so at just about 100 megahertz, and I think that says 165 megahertz. It's too small. I should have made those bigger. We start to see these striped patterns show up across the surface of the microtubule that may be a topological feature showing up. His work also showed what he calls the triplet of triplets, speaks of often in musical terms, which is a multifractal resonance spectra, where he's able to see, looking at just tubulin and exciting them in a two-dimensional way, we get these triplet of triplet structures, where we have 3 excitation peaks, and inside of each one of those we have 3 excitation peaks, and if we zoom into those, there's another 9 excitation peaks inside of each one of those peaks. And these appear whether or not we're looking at tubulin, we're looking at an entire microtubule, or we're looking at an entire neuron, we get this same fractalline excitation structure. This multifractalline excitation structure is a signature of quasicrystalline materials, where on the left we're looking at a simulated multifractal density of states of a coarse-grained Fibonacci chain, where the Fibonacci chain is the simplest one-dimensional quasicrystal that's used for a lot of these kind of studies, looking just at basic electronic properties. So this started to suggest to me when I came across this, that microtubules themselves and tubulin themselves may be some sort of quasi-crystalline architecture, which also seems to appear in the geometry of the centriole, as the centriole is clearly a very geometric, symmetric structure, but it has non-crystalline symmetries, as its central symmetry is 9-fold, and the microtubule symmetries are 13-fold, and neither of these have any sort of ability to create a crystalline lattice around them. They're classically forbidden. However, you can have quasi-crystals with these as your central symmetric points. And looking at Anirban's work onto the actual resonance spectra, there seems to be this growing body of work to suggest that the cytoskeleton is quasi-crystalline in nature. This is also in line with some of Hameroff's early work on the cytoskeleton, where he was looking at Fibonacci lattices in the microtubule. Where if we look at this, these are, what do they actually use? Well, here we're looking at the diffraction pattern on top. I'm forgetting the technique that they use to create these models of their microtubule. But what they're able to show is that in the tubulin lattice, we have, for one complete rotation, we either take 3 steps, 5 steps, 8 steps, or 13 steps, which is the Fibonacci sequence. And this just sort of shows how the Fibonacci sequence is directly built into the microtubules, which is sort of our one-dimensional quasicrystal already linearly, and then potentially a 3-dimensional quasicrystal coming out around centriolar structures. We also see quasi-symmetry in the potential predecessor to the centriole, as viral capsids use icosahedral quasi-symmetry in order to assemble themselves. And this pan on the wall here I have, the reason I have it is it's entirely coated in icosahedral quasicrystals, which were briefly used by a French company in order to create a non-stick surface, as almost no other material in the world that we come across will have quasicrystalline lattices. Nothing sticks to this guy. So we have icosahedral quasicrystals on here, created through some very specific stoichiometries of aluminum, iron, copper, and chromium, I believe. That's just an aside, though, is that in the earliest viruses, these quasicrystalline symmetries allowed themselves to assemble into highly ordered structures with the minimal number of essential parts that potentially, seemingly more likely every year now with the advancements in giant virus research, which have only been really coming out for the past decade, but have potentially gone from a icosahedral quasicrystal into a more complex quasicrystal in the centriole that, similarly to the modified icosahedral viruses that we see here, that when we add these extra fluorophores on the outside, we get these super radiant quantum mechanical effects. This appears to be similar to the icosahedral virus associating with the tryptophan-rich microtubules in order to get these distributed super radiant quantum mechanical effects on their own lattices. Now, that takes us pretty much to the end of this presentation. Do we have any questions here before we briefly talk about water structure?
Dan, do you have any questions? I have a couple.
Go for it.
Yeah, the cross-section of the neuronal axon, that is really interesting because if the axon was just meant to convey electrical information, you would intuitively imagine that the structure would be more similar to like a plant root, following an organic path, breaking, twisting, just finding an optimal, good enough path. But the fact that we see inside of the axon this very structured linear path of the microtubules, to me, makes, this is my question for you, does it make you think that, okay, this actually provides further evidence that there's something else besides just an electrical current, which might be the classical interpretation of axonal behavior? Like, why so much structure there?
To me, it almost reminds me more of just a close packing, where if I got a bunch of cannonballs or oranges and put them in a box and shook them until they got into the close packing, you're going to get this same hexagonal lattice for that reason. And all the microtubules, even though they're not touching here, they're highly charged objects. So they have got lots of negative charge on their outside. So they'd be feeling that charge as well, sort of to separate them into this optimal close packed system as the membrane sort of pulls them together. So that's my intuition on that one.
Yeah, that makes total sense, close packing. Now, regarding the infrared sensing in the 1990s via the Centriole, has there been any follow-up work on that? Because that is incredible, and that's also some related to some work that Dan and I are interested in chinchillas and insects being able to detect for infrared light, and where that might come from in the cytoskeletal structure, like in the dendrites of insect chinchilla. So I don't know if you have any more information there.
Yeah, unfortunately, very little follow-up. Albrecht Buehler, he published several well-regarded publications on this. He was able to demonstrate that it exists. He was not able to show where in the centriole would be absorbing this, though. And at this point, I believe he's retired. And very sadly, his website recently came down from online, but it is available on the Internet Archive. So if you need a link to that, there's a wonderful website going into his whole concept, his whole idea as a cell of an organism, where he views the centriole as the eyes, and as the eyes are directly part of our brain, their central nervous system, it's the eyes and brain of the cell. The microtubules are the nervous system. The actin is the musculature, and he goes into the entire cell, understanding it as a scale invariant organism.
Yeah, two comments there. First, the centriole is the window to the soul. Second, we need to archive their stories and websites while there's still time.
Definitely. Yeah, bless the Internet Archive.
Blessings.html to them. Okay, continue though.
Sure. So now we'll go briefly into the concept of not just the centriole, the cytoskeleton as a quasi-crystal, but potentially the cytoplasm of the eukaryote as a quasi-crystal. So what we're seeing on the left is a model for quasi-periodic solid water that was developed several years ago. And on the right, we're seeing a traditional quasi-crystal from the wall of a mosque, where this is a strictly quasi-crystalline tiling. And the way we can say it's really strictly quasi-crystalline is it has an expansion rule built into it, which allows us to subdivide the tiles further. Whereby how they showed this is we see the large line systems creating large tiles here, and then smaller tiles inside of them subdivided. So we could continue zooming into our tiles and continue subdividing them forever to get this highly ordered quasi-crystalline structure that has rotational symmetry around its center point, but no translational symmetry, which is what differentiates our quasi-crystals from our crystals. And then I've also chosen several viral capsids there that perfectly line up with this illustration. So we have our three different sizes of viral capsids sort of overlaid onto this as well in this illustration. But briefly talking about water structure, water in the cell is in a highly confined state. So in the very center there, we have an illustration by David Goodsell, who is the head of communication and public education at the Protein Data Bank. It's his job to provide the best illustrations of proteins available to biologists, and this is his take on cellular water, where we can see there's a very few water molecules between our proteins, which are these highly charged surfaces that sort of order the water on that. We also see that in protein collectives on the left, as our proteins start to get closer together, the water really starts to slow down, which is showing us it's entering into a more ordered state than bulk water like I have in my mug here. And, yes, or in there. And then on the right, in these really tight gaps between nucleic acids in proteins, or proteins, nucleic acids, our water acts more like a glass than a liquid, where it has really significantly slowed down motional dynamics. Now, water is a very interesting molecule, and a lot of its special properties come from its tetrahedral coordination, where water forms these perfect little molecular tetrahedrons with its hydrogen on one side and then its free electron pairs on the other side. So we get this little polarized tetrahedral element. And tetrahedrons tend to stack in very interesting ways. They come together in ways very different than spheres do, where most liquids can sort of be approximated by interacting spheres. This doesn't work at all for water. In order to really simulate its properties, we need to think about this tetrahedral nature. Now, this is just looking at how the golden ratio appears in the icosahedron. But we are getting to an icosahedron here. That tetrahedra very easily assemble into icosahedral structures. And this sort of close packing of tetrahedra is known to occur in water, where we get these little clusters of icosahedra that are known as flickering clusters in water. So even in your bulk water, you'll have little flickering clusters of tetrahedra that form and then dissolve, and they'll come together and come apart, and come together and come apart. So this is sort of just locally stable structure that's lower energy than just a random arrangement of tetrahedrali coordinated molecules that allow it to form, but due to the heat and motional dynamics of water, it tends to quickly break apart. Now, the icosahedron has long been associated with water. We can see here in the platonic solids, the icosahedron was associated with water, where tetrahedron was fire, hexahedron with earth, octahedron was air, dodecahedron was typically associated with the ether, and that is from I believe Plato did all of the others aside from the dodecahedron, and then it was Aristotle who added the dodecahedron for ether. But the icosahedron has been associated with water since the time of the ancient Greeks, which is now known to be a real state on the molecular scale of water. Where on the right, by the way, these are just not really associated with the presentation, but just quite fascinating to me that all of the platonic solids appear in the algae. Quite gorgeous. But the icosahedron has been associated with water largely due to the appearance of these fivefold structures in across the world of biology, in flowers and plants, in animals, in man himself. We see these fivefold symmetries, as well as the fact that if you take a handful of icosahedrons, they flow. Icosahedrons, they tend to roll, they flow very similarly to water, which is what I'm showing here. Now, it's also icosahedral cells having this fivefold symmetry. They can't pack into a closely packed crystalline symmetry in any form. And the icosahedron, another reason it was associated with natural forms is due to its close correspondence to the golden ratio, where you can form an icosahedron by three mutually perpendicular golden rectangles. They'll form the points of your icosahedron. And here we're also seeing how you get to the dodecahedron from the icosahedron, how they really share a lot of the same symmetries. And in viral capsids, you'll actually see both of them showing up as well in sort of in-between points. So below, you'll see all of these in-betweens between the icosahedron and dodecahedron. You'll find all of those in viral capsids as well. Now, quasicrystals also have the ability of forming icosahedrons, which is a non-crystalline arrangement. You could never form a true crystal into an icosahedron. quasicrystals, you can grow these metallic quasicrystalline icosahedrons with a local icosahedral order. And on the left, we're seeing how this works. How you can't close pack your icosahedrons, but if you overlap them slightly, then you can grow them onto each other using your quasicrystalline growth rules. Now, this is what's occurring in metals, and is what I have on my pan right there, is these icosahedral metallic quasicrystals. But this same thing is possible with our little icosahedral water unit cells as well. By overlapping our little water icosahedrons, we can grow larger and larger quasicrystalline structures. It's just that they're not favored in bulk water due to thermal motion and the tightly ordered networks that they would require. Here we're looking at a bit more the metallic glasses as well, where glasses in general, the metallic glasses are really closely related to the metallic quasicrystals. And in metallic glasses, you'll normally have these interpenetrating grains that have locally quasicrystalline order. You have these interpenetrating grains of sort of quasicrystalline overlapped icosahedra, as we're seeing below, that have disordered networks sort of between them. This is how it relates to some other Penrose's work, where Penrose, very well known in the quantum biology world, he's also better known in the mathematical world for discovering quasicrystals for the Western world, at least, where quasicrystals were known by the ancient Islamic architects and the general world of Christians and Hindus that surrounded them as well. The quasicrystal was sort of passed down as an artistic artisan trade. It wasn't until Sir Roger Penrose that they really entered the world of mathematics and physics through his independent discovery of them. Here we're seeing more of the subdivision rules associated with the Penrose tilings as well. As well as where the golden ratio shows up within his tilings. And his tilings are actually essentially a two-dimensional cross-section of a three-dimensional icosahedral quasicrystal, which is what we're seeing here, is how his tiles can be used. If we take a two-dimensional slice of our real three-dimensional icosahedral quasicrystal, we get the Penrose tiling showing up with all of its golden ratio based structures built into it. Now, this is potentially related to the golden ratio based structures showing up in the microtubule as well. Now, this is just a different ways to interesting ways to form quasicrystals, or we'll get back to this slide later. Let's talk about the water for another second. Where if we look closely at water, we spoke about how these very small icosahedral water clusters form all the time in bulk water. We also get slightly larger icosahedral water structures. So this right now you're looking at is a simulation of a oscillating icosahedral water structure. So it's sort of equilibrium state at a higher temperature. So it's a non-frozen state. And these have also been experimentally observed. They occur in very flickering, just really tiny amounts in certain bulk water samples, but they can be induced to form when you build a sort of like a cage-like structure that has the right symmetries. You can induce a larger icosahedral water cluster to form. So that's what's going on in this. In organic chemistry communications, they were able to form and demonstrate that they had a significantly larger icosahedral water structure inside of their cage molecules. And these icosahedral water clusters can just continuously grow into quasicrystalline water architectures. Now, these architectures are based on several different coordinated polyhedrons in the base level. which are very closely related to the more common clathrate hydrates. So what we're seeing here are crystalline arrangements of water and typically gases or small molecules that form these crystalline structures that, because of the tetrahedral nature of water, often have icosahedral local elements packed into a larger crystalline lattice. The phases, on phase diagrams between quasicrystals and clathrate hydrates, they're right next to each other. And this Nature Communications does a nice job of talking about that as well, how closely related these two forms are. That clathrate hydrates are essentially taking a section of a quasicrystal and then repeating that in a crystalline way, typically. Now, this is on studies of hard tetrahedra, so simulations of a pure tetrahedra, which are often used to simulate water as well. That water itself acts very much like a fluid of hard tetrahedra. That many quasicrystalline phases have been discovered in simulations of hard tetrahedra. Several were discovered in directly simulations of water in very thin layers. So in layers of around a nanometer gap, in simulations at least, extended quasicrystalline water phases have been discovered. But what's stopping larger quasicrystalline water structure like this from being stable is temperatures and the fact that there's no real boundary condition to enforce this structure. However, in the icosahedral viruses, we get this boundary condition that would enforce this structure of water, which is very similar to the actual, real icosahedral water clusters that were experimentally reported inside of icosahedral cages. The interior of viruses may contain this quasicrystalline phase of water as well. So I'm currently working with researchers. This idea only came to me two months ago now. But I'm currently working with some researchers. We're trying to figure out if we can look at viral capsids, ideally strip anything else out from the inside of them, and see if we can see these icosahedral water clusters forming inside of them, as we now have boundary conditions that would enforce this phase of water. Here we're looking again at a icosahedral virus. If we slice it along its center plane, we actually get a dodecahedral symmetry as well. So this is 10-fold. While we sliced it vertically, we would have gotten that 12-fold symmetry. We get the 12-fold symmetry here. But if we slice it along its middle plane, we get 10-fold symmetry. That's also a 10-fold quasicrystal. So all in all, this is a potential reason, a potential driving cause for the quasicrystalline elements that we're seeing in the cytoskeleton, that it may have emerged directly out of much more ancient cytoplasmic water quasicrystals from our early viral world. And that's most of what I have for you guys today.
That is absolutely fascinating, Daniel. Really good luck with this experiment, looking at
Thank you.
Structured water inside the viral capsid. Just one detail curious about: how do you get eyes on water inside of a viral capsid through the proteins so you can experimentally prove if they're there or not?
Mm-hmm. That's the difficulty.
That's the rub.
Yeah, at the moment we're looking at more indirect tests of this. So we're looking at ion transit inside of the cell, and I believe the scientist that I'm working with, it was his idea, ion cyclotronic resonance, where we look at the resonant patterns of ions and the sort of paths that they take. And in more coherent domains of water, we expect to have much larger paths. So we're planning on taking unstructured viral capsids with water inside of them, look at the passage ions form, and then these icosahedral shells, and seeing if it drastically changes our ion cyclotronic resonance. So we'd have both of them viral capsids, some of them unstructured, some of them icosahedrally structured, and see what differences we get from the water inside of them.
Yeah. Another question related to this, you know how they can super freeze water to get it in a supercritical state to try to prevent it from crystallizing, freeze it very quickly, and maybe you could crack the little virus open like an egg. I don't know if that's possible, but just riff on that.
That's a good point. Actually, I mention, yes, amorphous ices and supercooled water. So when we want to freeze a biological sample and keep all the molecules essentially where they are, you need to supercool it, just as you said. And what you're trying to do is prevent the formation of a crystalline ice. And you plunge the water directly into this amorphous ice, which is a glassy state of water, which is just like the metallic glasses we spoke about. How the metallic glasses are formed by interpenetrating quasicrystalline grains. This amorphous ice also appears to be formed by these interpenetrating grains of overlapping icosahedra, just like these quasicrystalline phases as well. So in this phase that we try to get our samples into in order to get biologically accurate images, we're sort of pushing it directly towards these local quasicrystalline pockets. And cellular water itself is obviously, if we're trying to get it to form this amorphous ice in order to keep everything in its place as it was while it was alive, cellular water is very closely approximated by amorphous ices, or supercooled water as well, which is quite close to this glassy amorphous ice form. Where the difference between amorphous ice and supercooled water is essentially supercooled water, we just get more motion, but very similar arrangements as to the amorphous ice. But you'd have, instead of being fully solid, you'd have a little transit along the rings, which is actually the same way that diffusion tends to work in quasicrystals, that each one of these little icosahedral units, all the atoms inside of it can rotate together. And you can have all these little wheels rotating to allow things to diffuse throughout it. And that appears to be what's happening in supercooled water, and potentially as well what's happening in our cytoplasm as well.
Beautiful. Dan, do you have any comments or questions?
Yes, Mr. Daniel, so many comments and directions. It really was a tour de force from the meta material to the meta aware. Even that last comment, wheel within wheel, hearkening to Blake and Ezekiel and all of this. Here's where all of that material together took me, though, was you began with that fundamental wave particle scale-free realization that even droplets of water and oil can exhibit quantized behavior. And it made me think about focused and diffuse attention and laser-like focal attention and more diffuse wave-like sentient forms. So here's my question to you, and it's not a gotcha. What is the nature of the scenario that we are in?
The nature of the scenario that we're in. So I'll take this as I understand it right now. I, myself, experience a reality. I know very little about it, to be honest. I make mainly assumptions. I assume everything around me is real, exists, fundamentally. That's a great assumption to make in order to act within a society. So it's a necessary one. I assume other creatures who look like me, act like me, are also conscious beings. Now, internally, myself, at this point in my scientific, my quest of knowledge, I believe that what I say is what I mean by I. Use entangled system that is mainly centered on the biological form that makes my body. However, in certain states of consciousness that I've experienced throughout my life, the exact boundaries of that seem to dissolve a little bit. And we can sort of blend with other creatures, other beings near us, friends, loved ones, close ones. So I guess, could you phrase your question again? Exactly what it was?
Yeah, just the overall nature of our experience and reality and situation, where we have a duality between focused and diffuse at the material level, but the material level itself reflected only through our interaction with it in a focused or diffuse way.
Mm-hmm. Yeah, so I suppose you're making me sort of think right now that what embodies us is our focus on being embodied. That if we take this sentient wave mechanical view, that in order to create these sentient waves that are our thoughts that interact with, by shifting our focus, by shifting our attention, we sort of shift where these waves are pushing the particles of the material world around us.
Yeah. Again, I really appreciated the presentation. I think it raises great questions. I love seeing the snapshot of your inquiry and how many threads are coming together there, and the tetrahedral water and the isomorphism of natural and sacred geometry, which are truly one and the same.
Mhm.
Yeah, I believe so. It's sacred because it's natural. We see it in the world around us.
Yeah, and it's natural because it's sacred. That's awesome.
Mm-hmm.
So, Dan, just
Yeah.
to wrap up, what's in store for you over the next couple months?
Yeah, so over the next couple of months, ideally we get some experiments going to look at the water phases. I am also recently involved with Hanson Robotics and James Tagg of the Penrose Institute. We're going to be trying to do some quantum biological experiments using brain organoids. So taking brain organoids and trying to recreate some tests that came out of the PAIR labs at Princeton in the past, where they were looking at, or as well as Daryl Bem's work at Cornell, which was all going on late 20th century. And they had very surprising, very interesting results that there was some sort of retrocausal influence on their experiments. But all of those experiments were on human samples, human participants, and that caused some doubt. So we're going to try to recreate those experiments in pure organoids, see if we can have a retrocausal influence within our organoids, or we see if we can take two organoids grown from the same pool of cells, two brain organoids, have non-local influences between them. So just sort of taking a lot of these foundations of psi-style experiments and taking them out of the human experimenter paradigm and putting them into the organoid paradigm. We'll be doing this, I believe, through, I know James Tagg has some of his own organoids. With Hanson Robotics, though, we're working with Final Spark. They're a Swiss company where you can rent organoids from them. You need to apply with your experiment ideas. They're interested in doing this, which I thought was fantastic, that they're interested in doing some quantum biological work. So yeah, organoids, water structure, and I'm still trying to find somebody to fund a ultra fast 280 nanometer UV picoquant laser. Where if we can do that, I can give it to Justin Karam, who is the wonderful scientist behind this work that we were looking at earlier with our super radiant synthetic nanotubes, where he's looked at super radiant systems before. He's worked on the biological photosynthetic systems that are super radiant. He's created the most super radiant organic materials known to date. And in order to extend these experiments into the cytoskeleton, we need a ultraviolet laser. So ideally, we can get that. We can start to do some of these super radiant experiments in the cytoskeleton and the centrioles and the cilia, and look for those quantum properties as well.
Fantastic.
Amazing.
Just a little, sorry, Dan, just one side note really quick. James Tagg and his partner Will, Daniel actually introduced me to them because they live in San Diego, and I've been to their little home laboratory and talked to them about their quantum experiments and their trials and tribulations getting organoids at UCSD. So it's a small world. That's so funny. Yeah. I'll text Will and James.
Well, I haven't met him yet. We're speaking at that conference I'm going to in San Francisco in 10 days.
Okay,
so
cool.
I'm very excited to meet him.
Yeah, it's all entangled.
Well,
yeah.
This was epic. Really appreciated the presentation, Daniel. And I will close with the final stanza of The Road Not Taken by Robert Frost. Here's what he wrote. I shall be telling this with a sigh. Somewhere ages and ages hence, two roads diverged in a wood, and I took the one less traveled by, and that has made all the difference.
Non-locality.
Eternal words.
Wow.
Wonderful.
Thank you, Daniel. Thank you,
Tucker. So much for having me on here, guys.
Yeah.
It's been wonderful.
Great.
We will stay in touch, man. Looking forward to hearing about your coming work, man. Good luck.
Peace.
Thank you.